Hello, everyone. My name is Federico Montealegre-Golcher. I'm a veterinarian and immunologist. Currently, I serve as the medical and scientific director for Nova Biomedical in Waltham, Massachusetts. Today's topic is surviving sepsis in veterinary medicine. That's Chapter 1, Development of Organ Failure. This presentation contains only educational materials that are free of any commercial content. For today, we have the following agenda: introduction, a review on the immune response in vital organ failure, neutrophils in organ failure, and a few conclusions. So let us begin with a small introduction on sepsis. After all, what is sepsis? Basically, it is a dysregulated inflammatory response to infection or trauma. It is progressive, organic, slow, and causing multiple vital organ failure, coupled with critical reduction in tissue perfusion. More than 1.5 million people get sepsis each year in the U.S. Close to 250,000 Americans die from sepsis each year. 7 to 10 patients with sepsis had recently used healthcare services or had chronic diseases requiring frequent medical care. And 1 in 3 patients who die in a hospital have sepsis. So sepsis has been redefined a total of 3 times. This is the last consensus on definitions on sepsis, and sepsis is defined as an organ dysfunction caused by a dysregulated host response to infection. And a shock is a subset of sepsis in which underlying circulatory and cellular metabolic abnormalities are profound enough to substantially increase the mortality in those patients. In this graphic, we have the annual sepsis incidence in the U.S. The important message here is that sepsis, septic shock, have been increasing on a regular basis since 2004. In comparison of the average length of stay for septicemia or sepsis in hospitals, we see this graph that on the x-axis we have the age, and in the y-axis we have length of stay in number of days. The blue bars represent sepsis, and the green bars represent other hospitalizations. As it can be appreciated, hospitalizations due to sepsis have obviously larger average length of stay in hospitals. For the financial impact of patients with sepsis, we can see that the ranking number 1, septicemia, is the most expensive conditions to be treated in the U.S. In veterinary medicine, we need more data on sepsis. Nevertheless, what is available indicates that at least 4% of dogs admitted to the ICUs are because of sepsis. In terms of mortality rates, we have different rates ranging between 50 to 36%. These are the publications available for the mortality rates in the last couple of years. So let us review the immune response in vital organ failure. A summary of the immune response. On the left column, we see several arrows that will start with stimuli, sensors, and effector cells. We have modulators and biomarkers, and we have outcomes. We will be dealing with sensors, and in the cells, we'll be focusing on neutrophils. Here we have the sensors. Here we have the organs affected, and obviously today we'll be focusing ourselves on the neutrophils and their role in organ sepsis, in organ failure. Just a review on the immune response. Basically, is divided into two major phases in systems: innate immunity and adaptive immunity. In innate immunity, we have the microbes, or we have molecules derived from tissue damage, that these will be sensed or detected in epithelial barriers by the neutrophils. And we have the triggering of several inflammatory cascades leading to organ failure. This is a very nice paper that describes the molecular biology of inflammation and sepsis. In this paper, we have two different origins of molecules that will trigger a response that is being sensed by three types of receptors. The molecules are those derived from pathogens or those derived from damage from our own cells. These cells, they have molecular patterns. So we have two types of patterns: pathogen-associated molecular patterns or damage-associated molecular patterns that they will be recognized by three types of receptors. These receptors are present in the cells of the immune system. We will be focusing on one of those receptors, which is the first one you see here, is the Toll receptors. What are the Toll-like receptors? These are transmembrane proteins expressed in cells of the innate immune system. They do recognize molecules in pathogens that are not observed in mammals and activate signal transduction pathways leading to inflammatory responses to destroy these pathogens. They are present in flies, in mammals, that is homologous to these receptors in flies, and they are designated as the Toll-like receptors. We have a total of 11 proteins. Here we have, we should focus on the lower part. It says pathogen and PEMS. Those are the receptors that we're referring to. Initially, we have two groups of molecules. On the left, you will see these are molecules derived from inflammation or tissue damage, and they are endogenous molecules released by tissues and necrotic area cells. And on the right, we have those molecules produced by microorganisms, exogenous microorganisms. These molecules have patterns, and those patterns are recognized by these Toll receptors. And as you can see here, you have downstream cascades being activated that are extremely pro-inflammatory. Essentially, when you activate these cascades, you will then produce a structure called inflammasome, which is responsible to induce the production of all inflammatory cytokines. The immune response, local and systemic in terms of the innate immune system, we begin with the bacterium, and also there is, as you can see on point A, we have a balance between systemic and local. On point B, local begins to increase its response, bacteria begin to grow, and then when we have a systemic infection is when the inflammation is spread out throughout the system. The balance is broken down, so we have the initial phases of sepsis. You can appreciate the basic response where you have two types of inflammatory responses. We have a hyperinflammatory and then an hypoinflammatory. The arrow just shows here where in these inflammation processes, the cytokine storm begins to take place. On point A, you have here in yellow, when the system begins to control itself, you have a controlled anti-inflammatory response, and you may reach homeostasis. On point B, this inflammatory response may be uncontrolled. However, there are some patients that they reach homeostasis. And on point C, you have an uncontrolled anti-inflammatory response. The patient cannot control it, this type of situation. And then you have that most of the patients, they do succumb to the inflammatory response. In this week, we show here several things. So the first of all, on the x-axis, you will see that you have a liberation of biomarkers, which you may assess at your time. These biomarkers are used for diagnosis, to modulate the therapy, or to measure the outcome. The yellow bars show where you have an immunosuppression in these patients. Even you can control the primary infection, you may have a secondary infection or a nosocomial infection. Then the patient succumbs. So now let's go to the neutrophils. Neutrophils are the first line of defense. It is the main cell type involved in acute inflammation and in some chronic inflammations as well. The main functions include secretion of molecules, migration, and phagocytosis. In fact, they secrete over 400 molecules. So dysfunctional neutrophil biology in sepsis, it leads to significant changes in sepsis that will contribute to the development of all secondary complications. So when infection begins, there is a mobilization, marginalization, and rolling to the endothelium, and addition and transmigration from the wall of the blood into areas where the neutrophils are needed. To illustrate that, on the left, you see that we have first mobilization and marginalization. Second, the rolling of these cells in the endothelium, and then the transmigration through the wall of the blood vessels into the areas where they are needed. So continuing on this neutrophil dysfunction in sepsis, we see here on number A, which is the normal endothelium interactions between the neutrophils and the receptors. It's normal. You see that there aren't any other, involving more particles, more molecules. However, on B, you could see that we have activated neutrophils. These may change in color just to illustrate some of the changes that they're suffering. They have going through, so they have a couple of yellow dots. These are molecules that have been released by all of these cells that cause a greater accumulation of these neutrophils. So when neutrophils begin to accumulate, they are following certain commands, which is first initiate activation, then use signals, and then they suffer or they go through important changes. So we have three relevant questions at this point. What changes are found in the neutrophils during the migration to the interstitial space? What will the neutrophils find in the interstitial space? What will these neutrophils do in the interstitial space? So let us look for the answers for these questions. First of all, what changes do we see in the neutrophils? they change their elasticity. Pro-inflammatory mediators are released by bacterial products, and they result in the increased leukocyte stiffness. Neutrophils become sequestered in the capillary beds, in organs like liver, lungs. There is a reduction in the leukocyte movements during sepsis, that this may contribute even more to the neutrophil sequestration in vascular and vascular occlusion, thus promoting tissue ischemia and dysfunctions of various organs. Also, these alterations between the endothelial cells and the neutrophils are altered. The profiles of adhesion molecules and its expression in neutrophils and endothelial cells further promote firm and neutrophil adhesion to the vasculature. Moving to the sites of infection is controlled by cytokines and by a set of particles called microparticles. Also, there's a deterioration of neutrophil migration in sepsis, which means that the migration is compromised due to the excessive release of pro-inflammatory mediators. Here we have just this graphic to illustrate the presence of particles. In the white squares, you can see that we have microparticles derived from platelets, microparticles derived from epithelial cells, and microparticles derived from neutrophils as well. These microparticles will be guiding the production of cytokines and also the movements of the neutrophils. Microparticles, let's see what are they. These microparticles are produced by endothelial cells, by leukocytes, by coagulation, and they will enhance the pro-inflammatory activities of the neutrophils. This slide we show a couple of the receptors present in the neutrophils, and at the bottom you can see an endothelial cell secreting endothelial microparticles. These microparticles will start to accumulate. Then we have activation of our resting platelets. We have the trigger of pro-inflammatory mediators as well. You begin to activate the classic cascade of complement, and then we have a very, very dangerous situation. Here you can see in arrows and in the white lines of the squares the formation of microparticles in megakaryocytes from a experimental model in mice. The next step is to expand the inflammation. So in the acute phase, neutrophils entering the circulation can spread inflammation to other organs, and as a consequence, you will be leading to an additional damage. In the late phases, there is a state of immune refractoriness with reasonable amounts of anti-inflammatory cytokines and specific inhibitors of cytokines. Remember that there is a phase where we have immunosuppression. The lack of maintenance of balance between inflammation and anti-inflammation breaks down. We have complement activation, and then we have an expansion of the inflammatory process. Additional steps are taken. Here on the left, you will see that the neutrophils, they begin to activate themselves and to liberate a structure called nets. Once this starts on a septic shock, there is an occlusion of these spaces, and you will have a damaged organ. But let us see what nets are. Nets are extracellular traps, and they are formed by the histones and DNA, and the role is to trap bacteria and to kill it. They also contain enzymes which are responsible for the killing of the bacteria. We show a couple of microscope pictures taken from an experiment which shows the morphological changes in netosis. From the top left, you see that the chromatin loses compaction. Next, the nuclear envelope breaks. Next, the nucleoplasm becomes homogeneous, and at that time, on the lower left, there is a rupture in the cell membrane. The chromatin is expelled, and netosis is formed. So what is at this point in time? In the middle, you may observe the nets, and the next to the right, you will see that there is a clump, if we call it that way, which is a thrombus formation. It includes RBCs, it includes platelets, and then we begin the process of organ failure. This nice micrograph shows that the presence of nets in activated neutrophils. The role of the nets is to trap and to kill the bacteria. This nice experiment, we can see, first of all, on the left, on the micrograph EA, these are non-activated neutrophils. They have very nice round shapes. On B, once they're activated, they begin to change the morphology. On D, you begin to see that they make fibers. On E, the nets are formed and they are expelled into the surrounding environment. So nets will trap bacteria. We have here an example of Staphylococcus aureus, Salmonella typhimurium. In these examples, the bacteria are trapped, and remember that there are enzymes trapped also in these nets, and the bacteria is killed. The last bacteria is Shigella flexneri. You can see that these bacteria are in the process of being destroyed. And yes, nets do exist in veterinary medicine. This is an excellent paper showing that the nets do exist in animals, in dogs. Here we have an experiment. We have 3 or 4 different concentrations of an activating agent. The top row is the control, and you can see it on the last column on the right, shows normal morphology, and the 2 of different concentrations on 6.4 and 31 millimolar activating agents. You can see that the morphology is changing and the nets are formed. There are also a couple of other publications on bovine and in cats as well. These are other publications in veterinary medicine that shows that yes, nets exist and they have the same functions as we described in mares in endometriosis, in dogs, and in bovine, especially in bovine mastitis. Solutions. Neutrophils are the host first line against invading pathogens. However, in sepsis and in septic shock, there is a paralysis of the response, resulting in the inability of the host to contain and eliminate infection. Now, neutrophils are important contributors to form plaques and thrombus, leading to defective perfusion and organ failure, as shown here on the last slide. Thank you very much.
The agenda for today will state the purpose of
we will do the introduction about biomarkers, lactate. We'll discuss a few things on procalcitonin, and then the discussions. So the purpose is to address the use of biomarkers in sepsis in veterinary medicine, with emphasis on lactate and procalcitonin. Let's begin with a small introduction. In chapter number 1, we discuss the immunopathology of sepsis, and then you will see on the first column you have certain phases that will begin with the insult, the trigger, sensor and effector cells, mediators and biomarkers, impact on organ function of those mediators and biomarkers, and an outcome. So we'll be focusing on mediators and biomarkers with emphasis only on 2 biomarkers, since there are over 175 biomarkers with some degrees of evidence today. So as you know, it is a time frame situation where biomarker assessment has to be conducted through time for diagnosis, monitoring therapy, outcome, surrogate, and stratification of patients. So let us begin a little bit on biomarkers. Markers is a characteristic that is objectively measured and evaluated as an indicator of normal biological processes, pathogenic or pharmacologic response to a therapeutic intervention. Usefulness is evaluated by the capacity of those biomarkers to provide timely information that is critical for each condition, beyond that is really available from routine physiological and clinical data, speed and accuracy. Then we have sensitivity and we have the specificity of these biomarkers. Let us review a little bit the literature to see what are we facing today. So there is a publication, a systematic literature review, and the methods were they searched the PubMed database using the keywords sepsis and biomarkers to identify clinical and experimental studies which evaluated a biomarker in sepsis. And the results show that there are 3,370 references covering 178 different biomarkers. So the authors reached the conclusion that biomarkers have been evaluated for use in sepsis. Prognostic markers in sepsis, they are relatively few, have been used for diagnosis. None has sufficient specificity and sensitivity to routinely employed in the clinical practice. So we have a classification of sepsis markers, which include cytokines and chemokines. We have, for example, interleukin-1, the receptor antagonists and TNF. Cell markers, we have the CD40 receptor, the Fc gamma receptor number 3, coagulation, activated partial thromboplastin enzymes, vascular endothelial damage, we have the platelet-derived growth factor, vasodilation, angiotensin converting enzyme. We have also organ dysfunction, the troponin in the heart, acute phase protein, CRP and calcitonin, and others which we include lactate. And there is a category by themselves which include troponin in the diagnosis. So what are the role markers for us in the clinic? Well, number one, it is to identify a patient with a high probability of disease, correct diagnosis, an adverse outcome, or a benefit from intervention. Identify presence or absence of pathologic state and processes. 8, in risk stratification and prognosis. Monitor response to an intervention or treatment, and serve as a surrogate and point in clinical studies as well. So let us begin with lactate. It's a conjugate of base of lactic acid, pKa low, 3.9, two optical isomers, L and D, produced endogenously as a product of anaerobic metabolism. And the only stress condition under normal conditions, it's about 0.5 to 2 millimolar per liter. So metabolism. At the top, we have the mitochondria, oxygen, that is the aerobic part of the cycle. We have the glucose metabolism under normal conditions. It goes through the aerobic pathway, enters the pyruvate, where we have the pyruvate dehydrogenase, goes to the citric acid, and pyruvate is produced, CO2, H2, and 2 lactate and 38 ATPs. That's our normal energy production. So we have the glucogenic pathway, glucose metabolism in times of stress, disease, exercises, accidents, trauma, infection. So we have high cellular ATP requirements. And that coupled to a mitochondrial dysfunction because they cannot put up with the demand, so they go for the anaerobic pathway. It is switched to a less efficient metabolism, which is the pyruvate, with the lactate dehydrogenase into the Cody cycle, which actually recycles everything back again. Nevertheless, in this point, we have only 2 lactates and ATPs. Now, there's a rise in lactate locally due to sustained tissue hypoxia, higher anaerobic metabolism, and decreased lactate clearance, all of them taking place at the same time. So lactate concentrations is a balance between production and clearance. Under stress, which already mentioned, there is a close to 0.5 to 2. And under strenuous conditions, exercises, and so on, we can reach with patients 14 up to 20 millimolar per liter. Now, why lactate? Well, under stress, lactate is a source of energy. It's a package in the same cell where it is produced and also in other cells where it can be used as an important fuel for oxidation and glucose generation. And remember that in trauma, sepsis, exercise, surgery, and so on, we need high levels of glucose generation. So the blood lactate is a marker of stress, and also it is important for us to consider lactate clearance, which is an independent predictor of mortality, targeting resuscitation in sepsis to achieve lactate clearance of at least 10%. Now, hyperlactemia is common in patients with sepsis, a marker of illness severity, and a strong independent predictor of mortality. The lactate levels, we have 2 types of hyperlactemia, A and B, and each one of them is subdivided into further subgroups. A has 2 subgroups, where we have increased oxygen demand, which is exercise, seizures, shivering, decreased oxygen delivery when the patient might be in septic shock, blood loss, and severe anemia. Type B hyperlactemia, not due to hypoxia. First, it could be associated to an underlying disease, such as sepsis, associated with drugs or toxin, acetaminophen and cyanide, ethylene glycol, methanol, and inborn metabolism errors. And we have here a series of mitochondrial myopathies and enzymatic diseases. In animals, we have, in mature dogs and cats, the following levels. In the first column, you will see the lactate concentration and the next, significance. So it starts with less than 2.5 millimolar per liter, which is a normal condition, normal levels. 2.5 to 4.9, mild elevation, 5.5 to 7, more or less, moderate elevation, and on the high end, above 7, That's a severe elevation. So how can we quantify the lactate? Well, there are laboratory methods for serial lactate measurements, and it's important to indicate serial lactate measurements. You have a central lab, could be more or less 30 minutes, more or less or more. It all depends on the patient load and sample management. They have enzymatic methods. Blood gas analyzer, you have, for example, more or less 30 minutes as well, depending on the patient load and sample management. At the high-end academic centers, you may have high patient loads. And you have the handheld devices, POC, point of care, which is less than 50 seconds, and they rely on enzymatic biosensors. So it is critical to have early measurements of lactic acid. Why? To allow a correct triage and address the patient to the proper hospital structure or pathway, to allow the proper therapy to be administered with no delay, that is, time to antibiotics and dedicated therapy according with the sepsis protocols, and to allow an efficient patient monitoring, whereas at the critical care unit. So delay impacts on patients' safety and costs of treatment, of course. So if we do a rapid diagnosis with point of care, this would allow a reduced turnaround time in diagnosis, maybe less than 15 seconds, reduced blood sample, maybe 5 to 7 microliters, improve key performance indicators of the patients, time to triage, time to antibiotics. So sepsis has an interesting cycle nowadays as well. You have a management flow of patients with sepsis. And in people, you have now many important clinical trials detecting lactic acid in pre-hospital identification of sepsis to rapidly channel those patients into the sepsis protocol. Here you can do with maybe POC and lactate. Maybe we can have that at the vet clinic, smaller clinics, that they're going to make a initial diagnosis and a patient assessment and run into a more specialized unit. Here at the vet clinic, they can do also determine the levels of lactate, and obviously in the emergency rooms where you have all possible equipments. In humans, it's very important nowadays to have all these studies conducted in pre-hospital care. So now let's see a few studies. One is early lactate-guided therapy in the intensive unit patients. This is a study in the Netherlands. It was conducted in 2006 to 2008. Lactic acid as a criteria for inclusion was 3.0 milliequivalents per liter. And they had two randomized groups, one with serial lactate measurements and the other one was the control. Here, patient flow, you can see more or less that they have two groups. On the left is the control, on the right is the ones that have lactate-directed therapy. These ones were subjected to serial dilutions and certain variables were measured. This is the format of that study, indicating that the lactate therapy favored early ICU admission, and this is critical for patient survival. All lactate monitor patients have significantly less length of stay and decreased mortality. Early lactate detection and serial measurements provide, obviously, clinical benefit. Now, we mentioned early on lactate clearance. So lactate clearance for death prediction in severe septic shock patients during the first 24 hours in intensive care unit. This is an observational study. These were 94 patients that took place in the study. You had, for each hospital, you have specific formulas for the lactate clearance. And the results clearly show that lactate clearance was higher in survivors than non-survivors for the 0 at 6 hours and the 0 at 24 hours. And the best predictor of death at day 28 was lactate clearance from 0 to 24 hours. We have here the area under the cuff, 0.79, with intervals 95% to 0.6 to 80.85. So in dogs, yes, it has been done. And the lactate concentrations and lactate clearance were good indicators of dogs with septic peritonitis. Now, we have this study that shows the results of 4 point-of-care meters that were analyzed for rapid determination of blood lactate in dogs. The conclusions of the authors indicate that the POC devices were useful in the clinic because it was very fast, obviously. When we take an example, it is important to know that lactate production by red cells continues to flow in blood. It means that we have to do it in less than 5 minutes. Maybe handheld analyzers are the way to go. Blood samples submitted to the lab analysis should be collected in tubes containing sodium fluoride. Solutions with 5% dextrose in norbasaline on Binger's lactate have been shown to alter results over time. And obviously, patients that are under stress and struggling can increase up to 10-fold, especially in cats struggling. This graphic shows that samples were placed on ice, and according with time, the levels of lactate were increasing. It is important to indicate that wet ice at 0 degrees decreased the production of lactate. So let's talk about calcitonin. That it's a lot of information nowadays in human, and it's getting into the vet field as well. The structure is a 116 amino acid peptide, and this is the precursor of the hormone calcitonin. This is the structure with the cleavage sites on the left and on the right. It is release of calcitonin in the context of the endocrine regulation. That is, the synthesis of healthy persons and animals. It is taking place in the C cells of the thyroid, and it means enzymatically converted to calcitonin and then stored in endocrine granules. And these are released under certain stress conditions. In the role of sepsis, this is an alternative cytokine-like pathway during the hormone chemokine. Bacterial toxins is very important here, gram-positive or gram-negative, and cytokines stimulate production of procalcitonin in the parenchymal tissues. This process can be attenuated or blocked during viral infection by interferons, non-endocrine tissue, for example, liver and lung and brain, since they don't have endocrine granules to be stored. And also, it's important to mention that procalcitonin is released into the bloodstream, but is very short-lived, maybe 12 to 14 hours. RNA expression in septic and control animals. In the first column, we have the tissues that the researchers analyzed. The first column, you see there is calcitonin and has a red square, and below it, you have control and sepsis. You'll see that calcitonin is expressed in almost all the tissues. TNF, almost very similar. Interleukin-6, very similar. And obviously, the control for the reaction beta-actin, control and sepsis were equal in terms of RNA expression. So in dogs, in this paper,
4 dogs were analyzed for infection, non-infections, inflammation, neoplasia, and others, and they evaluated the prognostic value. This graph. On the left, you will see a graph indicating the sick dogs in red and healthy dogs. There is an important difference, although a little bit overlapping, but important difference. On the right, we have 4-box plot. You can see that at least in neoplasias, in sepsis and inflammatory diseases, they look very much the same. So how we detect procalcitonin? By enzymatic immunoassays, ELISA, chemiluminescent, and immunofluorescent time-resolved amplified cryptate emission trace, which is a very, very sexy technology, if I can call that, based upon non-radiating energy transfer from donor molecules. This is uranium cryptate in red below, to an acceptor molecule on the right, and that excites the electrons, and then you can measure the reaction. Now, this graph shows a couple of biomarkers, and we would like to focus on procalcitonin. That is the orange line. Clinical symptoms alone are often insufficient for early and accurate diagnosis. Unless you have tremendous degree of experience, we need lab as well. The PCT levels can be observed then 3 to 6 hours after the infection's challenge, with a peak up to 1,000 nanograms per ml. That is after 6 to 12 hours, and then it drops. And it can be said from these results that it's specific to bacterial origin of infection and reflects the severity of the infection based upon the bacterial origin. We have the Kleeman assessment on accuracy. We have the orange points for procalcitonin. Now, the levels in this study accurately differentiate sepsis for non-infectious inflammation. And it has been demonstrated that the best marker for differentiating patients with sepsis from those with systemic inflammatory reaction, not related to infectious cause like trauma and burns and poison. Clinical relevant levels. On the first column, we have the different levels for people. Next is the recommended management, dealing only with antibiotics. According with the levels, you suppress antibiotic or you continue the therapy. And in dogs, well, there is a kit, ELISA, and this author's flora indicated that it's not very consistent. In horses, the procalcitonin was detected by ELISA and is helpful to detect in horses systemic inflammatory response syndrome. And also, it is more important in equine chronic pneumopathies. So conclusions. We have this graph, which on the top we have SIRS, sepsis, and septic shock. On the y-axis, we have concentrations of procalcitonin and lactate. And on the x-axis, we have these two biomarkers. They measure different biomarkers with different purposes. There is evidence that in lactate, management according to measurements improves survival. Conclusions on lactate. Lactate levels are a useful marker for organ dysfunction and serve as an endpoint resuscitation for patients with sepsis and sepsis shock as part of the bundles. In 2016, the sepsis levels were included in defining only patients with septic shock. Serial lactate measurements are useful in monitoring effectiveness to various therapeutic interventions, and therefore it is recommended in the sepsis bundle to septic shock, especially when the initial level is high. Monitoring the clearance is critical through serial dilutions, maybe 8, 6, and 12 hours, just to see how all the bundles are performing. Elevated levels of lactate are not considered specific for either diagnosis of sepsis or predicting mortality, unless we couple to the clinical picture and other laboratory analysis. Point of care determinations results in 15 minutes. They're very easy to perform, small volume, and good correlation with the central lab for critical care. Calcitonin is the only analyte approved by the FDA in the assessment of risk in developing severe sepsis in critical ill patients. But it is on the first day of admission of intensive care. Now, severe sepsis is no longer part of the sepsis definition. Lactate and C-reactive protein are both approved, but not specifically to sepsis. Procalcitonin is closely associated with inflammation, but not completely specific for infection. Evidence has shown that the elevated number of disorders in the absence of infection, especially following trauma. Using a single concentration value may not be practical at all. Procalcitonin should always be interpreted carefully, because remember, both biomarkers, they are telling us different vocabulary. And the immunoassays take about 15 minutes or more to produce results. As the closing, it's nearby. Sepsis management, we need to include biomarkers plus blood gas analysis, blood gas analyzer, and we have to follow up that patient. Thank you, everyone, for your attention. Now, if you have any questions, I'll be glad to answer them. Please go into the question and answer session. There's a top box in there that says questions. Type in the questions, and I will respond to them as much as possible.
Federico, you have one question. Is it realistic to use procalcitonin in a veterinary setting given the cost and time for analysis?
In my clinical view, may not be a good choice unless it's for equine medicine and respiratory diseases. It takes about 50 minutes and is very expensive.
The only other question we have currently is, can you please supply the reference for slide 31?
I sure will.
So we can provide the reference for you through email. And that looks like all the questions. So I'd like to thank everyone for joining. Wait a minute, one more question.
Thank you very much.
One more question, Federico. Where is the point of no return when you can confidently tell an owner that there are no good outcomes?
Well, it has to be a limit. You have to establish a limit with the owner, expenses, parameters, and be realistic on the patient's survival. Usually, it's a very difficult decision because it has a lot of emotional variables. Nevertheless, in my opinion, you should have a frank discussion with the owner, tell them the options, and have this person just make the decision.
There's one more question. What is the average cost of procalcitonin testing?
It would depend on the lab. Normally, I would say between $300 and so, but I am not sure since prices vary a lot. All I know, it is a very expensive test.
That looks like all the questions. So thank you, everybody, for joining.
Thank you very much.